order to familiarize yourself with the equipment that is used throughout the semester in a nutrition lab, we have put these simple videos together. Several common lab items will be presented here, and it is all included in the equipment identification assignment. Lab safety is taken very seriously in the nutrition lab, and the use of safety glasses, safety goggles, and gloves are required in several labs. During the labs, there are hazards such as boiling water, glass equipment, and chemicals. For this reason, closed-toed shoes are required for every single lab. Safety goggles should be worn by individuals who need to wear prescription glasses or readers, as they are not a replacement for safety glasses. Those who do not require prescription glasses or readers may wear safety glasses. Gloves are also available in sizes extra small through extra large, and they should fit snugly like this. Any extra slack may interfere in the dexterity or the manipulation of equipment during an experiment. In the portions lab, we will be using kitchen scales. They're used to weigh food items in either ounces or grams and to measure our different nutrient densities. In our blood assessment lab, blood value data will be collected using different apparatus. These items are used to collect the blood samples. There's a single-use lancet that is used to prick your finger, and then a small amount of blood is collected with this capillary tube. After the capillary tube has the collection, there are 3 different test monitors that are used. We have one that tests our blood iron, a second that does a full lipid panel, and a third that does our A1C or our blood sugar values. These data are all applied to your assignments in the blood glucose lab as well as the blood test lab. Anthropomorphic measurements are also collected by stepping on the Tanita scale. The scale provides a printout of the results, including body fat percentage, total body water, and bone density. Certain body measurements are also taken during this lab and applied to the body composition assignment during the test lab. In our taste lab, nose plugs are worn in the experiment to test your sense of taste without sense of smell. In a different experiment, these taste papers are used as indicators for a genetically predisposed taste determination. Beakers are used to hold solutions for other taste experiments, as well as multiple uses throughout our lab assignments. Beakers can be used for boiling water in a water bath or to hold hot items instead of solutions. The plastic ones are often used for waste containers. During the taste lab, solutions are placed into the mouth with disposable transfer pipettes, such as these. The basic skill of serological pipetting must be learned and practiced prior to the macronutrients lab. The equipment used in this exercise includes a serological pipette with a pipetting wheel. Using this assembly, solutions, such as this one in the flask, can be removed and placed into test tubes. These test tubes are secured with a test tube rack, and when samples have been processed and are ready for removal, or if they're too hot to touch, we can use test tube clamps. The sides of the test tube clamps are depressed from the outside moving inward, and then the grip is relaxed. Once the end is around a test tube, you can move the sample simply by lifting it and transferring it back to the test tube rack and opening again once it is in place. Reagents can be measured using a small graduated cylinder. It is more accurate than a beaker or a flask. When it comes to measuring liquids, reagents can also be applied to samples using a dropper bottle, and we have different types of reagents that are used in each of our labs. Sometimes a test tube is too large for a given substance, and that's when the experiment is carried out using a spot plate. Instead of using large serological pipettes in these small wells, solutions are provided in smaller 50 millimeter tubes and applied with the transfer pipette. Learning the skill of micropipetting is necessary prior to engaging in the Bradford assay. There are detailed instructions when handling the micropipettes, and depending on the pipette model or the size of the micropipette, different tips must be used and determined prior to use. Since the micropipettes are for micro amounts, meaning amounts in millions of a liter, the size of a sample tube is much smaller than a standard test tube. Here are some of the microcentrifuge tubes, and the maximum volume that can be held by these tubes is 2.0 milliliters, or 2,000 microliters. They are held secure by a microcentrifuge tube rack. The Bradford assay is an experiment that determines the unknown amount of a protein in a sample. After samples have been prepared for the experiment, they are analyzed by the spectrophotometer, and the collected data is plotted onto the graph to determine the outcome.